Hello, good morning students. Myself Hemanta Nayak and my channel is Nayak's Biology. Today we are going to discuss about the monohybrid cross and incomplete dominance. The most important is that we have to see the difference between them. We know, everyone knows, because 10th class also you read about the monohybrid cross. But now in 12th class we have a new concept, that is incomplete dominance. But what we will do, we have to understand what is the difference between them. First we will see the parents generation, both of them. Parents generation, what is the parent generation of the monohybrid cross? If you see the parent generation of monohybrid cross, it is TT, we have to cross with the small tt. Then now we have to understand what is this tt. It is the allele. What is the allele means? It is the different types of gene or different forms of the gene. And why we are writing not tt triple t, but because this allele only having the two allele which can express a particular character. Like that, because the T is a dominant, because it is a capital letter, we have to represent as a dominant. It is a tall. And small t, it is a god or short height. After that, we have to see the parents of the incomplete dominance. In the incomplete dominance, if you will see the parents, capital R cross with the small r small. What is this cross? Capital R, it is represents the red color, whereas the small r small represent the white color. What is this white and red color? It is just flower of the snapdragon, where it is the height of the Pisum sativa. Now we have to gamete formation. After the parent generation, you have to know about the gamete formation. If you are unable to know about the gamete formation, how we will combine them? That's why now we have to gamete formation. In the gamete formation, what it will, here it will produce the, because it is a homozygous condition, that's why it is only for one type of gamete. Now it is capital T is the gamete formation here, and here it is also in homozygous condition, it is the small t, and we have to cross with them. Then it is comes capital T small t. Here you see it is just RR and small rr. Here also it is homozygous condition, it will produce the R, and here it is produce the small r, and you have to cross with them. Then you have to cross with them. Then what is the combination it came? Capital R small r, here capital T small t. Now it is in the heterozygous condition. What is the meaning of heterozygous condition? When the two gametes are, two different gametes are. You see here capital T is there, here small t is there. Capital T is there for the dominant character, whereas the small t is there for the recessive character. Now we can tell it is a hybrid. Why it is called as a hybrid? Because it contains the different gene. And what is the its phenotypic? What is the character? It is a tall plant, because this capital T is dominant over the small t. Because it is a, this character of the capital T is dominant over the small t. That's why it is a hybrid, and its height become tall, because here T is dominated. Here, but the things are different in the snapdragon and incomplete dominance, because it is a red and it is a white. When they will combine, when we will cross them in F1 generation, then the color will come the pink. Why it is a pink? Because simple logic you have to use here. The red color, if you will mix with the white color, the pink color will come. That's why it is a pink in color. Why? Because the red color is unable to dominate over the white color. That's why it comes the pink. That's why the name is incomplete dominance. Now we are going to, it is a F1 generation. What you find? In F1 generation, we find that there is a, in monohybrid cross, we having the hybrid tall, although it contains the small recessive gene, but it is able to dominate its character. In the incomplete dominance one, in F1 generation, although red is unable to dominate, and that's why it has become the pink color. And now we will see about the F2 generation. For the F2 generation, what you have to do? For the F2 generation, we have to do self-pollination. What is the self-pollination? Suppose in a bisexual flower, what will happen? You know, bisexual flower, the anther having and stigma. Anther will produce the pollen grain, and stigma, what does it produce? It doesn't produce anything. Pollen grain will fall on the stigma and pollination takes place. It is in the same flower it will happen. That's why we are making it the cell pollination. In this cell pollination, what you will get? You have to cross the same gametes. We have to cross the same alleles, because in the same flower it is a male, consider it is a male and it is a female. When you will consider it is a male and a female, then automatically the cell pollination will be occur. In the incomplete dominance also we have to same thing we have to do, because it is F1 generation as we know, and we got the pink flower, and we have to cross it, that is by the cell pollination. What it will do, what it will come in, simply we have to cross it RR with RR, because it is cell pollination, same flower, the stigma and fall on the pollen grain fall on the stigma. Here the F2 generation, it is F2 generation. Now how we will know that what is the offsprings will come here? We have to make a simple table and we try to understand. It is all about the concept before the going to the F2 generation. I will recall it. It is a tall, it is a red color. Tall is dominant here F1 generation, and red color is unable to dominate its character. That's why it is called incomplete, and it is called the monohybrid cross. In the F2 generation, we did the cell pollination. Both of. Let's see what will happen in the F2 generation. What to happen in the F2 generation by a cross. Are you clear? Now we have to see about the F2 generation. What you did? We have to capital T small t crossed with capital T small t. Whereas we cross in the capital R, capital R, small r. Then simple, sometimes we are doing the cross like that. You no need to do ray. We have to, what you are doing, we have to ray like that. But no need to do, because it is getting somewhat confusion. We have to simple make a cross. What is this cross? Simple. It is a simple, very simple cross. In the genetics, you are very familiar with this. What you will do, it having the two gametes, capital T, small t. Let's it is considered it is a male. For the male, we have to make a sign here. And for the female also, we have in the same gametes, capital R, small r. These are the capital, it is T, capital T, small t, capital T, small t. Here what you have to do, same things we have to do without changing anything. You have to make a box like this, but gametes are different here. It is capital R, it is small r, it is capital R, and it is small r. And it is considered what you will, it is considered as a male and it is considered as a female. Here also same, here also same. Now we have to simple cross it. T crossed with the tree, that is T, it will come. T cross with small t, here it will come. Here capital T, small t, here it will come. And small t, it will come. Here what you have to do, same things we have to do. R we have to come. R cross with the small r. This capital, this is capital R. Here R small r. Here R crossed with the capital R and small r. Now we have to see about the phenotypic and genotypic condition of the monohybrid cross and incomplete dominance. Are you clear? Until now what you did, we did F1 generation, which it came the hybrid. In case of the monohybrid, there we got the pink color. F2 generation, we did the cross. Now we have to discuss about the phenotypic ratio and genotypic ratio. What is the phenotypic ratio and genotypic ratio of the both? Now, what is the phenotypic means? Physical appearance, how it looks. It is called the phenotypic. Genotypic what? It is genetic constitution, like that. Suppose a plant is tall. What is its character? It is phenotypic character of the plant. Genotypic means what is its gene? Maybe it is a capital T or it is a capital T small t. It is its genotypic. Now you will try to understand how many tall plants are there. Here it is a tall. This one is a tall plant, pure tall, because it is contain the two pure gamete. But here it is hybrid, but it is a tall because tall plant is dominant over here. Here also tall plant is dominant because capital T gene is the That's why we having the 3 plants are become tall here. And what is the one color? What is this? This one becomes short or dwarfed, because the phenotypic ratio is 3:1. Simple. What you understand in this? Suppose in the field 4 plants are there. In that 4 plants, 3 plants are become tall plant and 1 plant become short-eyed plant or dwarfed. Now we have to see the genotypic ratio. Genotypic ratio, how many, calculate how many capital T are there. In this cross, the capital T is only one. Genotypic means genetic constitution. What is the genes they contain? The allele, you know, gene means, allele means different form of the gene. This is the 2 genes, only this allele are having 1. But capital T, small t, it is different, then it is a pure and it is a hybrid. That's why it is 2, 1 and 1. And small t, how many it got? Only 1. That means that there is a different type of genetic constitution we find in the F2 generation of the monohybrid cross. And what is its ratio? 1:2:1. Is it clear? I will repeat. If when we have become phenotypic ratio, it is a physical appearance. Suppose in a field, 4 plants are there, 3 are become tall and 1 is short. Physical appearance, how it looks. But when we discuss about the genotypic, what is its genetic constitution? Capital T, 1 it came, capital T, small t. It is a different, although they have been both are tall plants, but genetically, allele, they having the different. And where the small t, only 1, that is 1:2:1. Now you will discuss about the phenotypic ratio of the incomplete dominance. Phenotypic ratio, what it will come? In this case, phenotypic ratio is physical appearance. Now we will discuss about the phenotypic ratio and genotypic ratio of incomplete dominance. When we will discuss about the incomplete dominance of phenotypic ratio, genotypic ratio, you see that capital R become red, whereas capital R, small r, it become not red, it become, here it is a pink, because here the capital T become dominant, or here it is not dominant. Now what is about the small r, it is become the white. Now consider it is a field, it having the 4 plants and 1 is red, 2 is pink and 1 is white. Then what is the phenotypic ratio? Physical appearance. If you will see the field, how it will looks? It looks that 1 it become the red, 2 it become the pink and 1 it become the white. Whereas genotypic, what you will write? Genotypic means what you understand? We understand that it is a genetical constitution of the allele. This allele, capital R, capital, only 1 we have. That is 1:2. Capital R, small r, what is its? It is 2. And small r, it is only 1. That means here it is phenotypic and genotypic ratio is same. That is what is the difference in the recapitulation. Only what is the difference? I will 3 points. Monohybrid cross, it is only in the F1 generation it is a dominant. For incomplete dominance, it is not dominant. It become the intermediate or intermixed color. That is pink. In the phenotypic ratio, it is in the monohybrid cross, it is 3:1, where it is 1:2:1. But whereas the genotypic ratio, both of the same. And thank you for watching my video.